at my place, it's actually not that late, but I bet that Dr. Sinha's home
I
it
know.
is very, very late.
We are on the same time
Oh,
zone.
oh, okay, okay, good,
Yeah.
good. So greetings again. Well, good morning to everyone in India. I have been attached to multiple institutions here in US, have been working on various type of work, including COVID also. Actually, there is just one paper came out today. And so I thought of what to present. So I'm very dear to vector-borne diseases, and especially neglected vector-borne diseases. So I thought of talking about it. So today what I will do is kind of give a brief outline of the broader work that I have been doing. And I will show some of the example studies from my research. So I'll be talking about the scaling, and of course, complexity hidden in these type of vector-borne disease. So complexity, what complexity I'm talking about, I will define some of the scales that are very relevant to the modeling of these diseases, and give some examples from the disease that I work very importantly is leishmaniasis, or Kala Azar, what it's called in India, in Bihar, West Bengal, and parts of Uttar Pradesh. And also Chagas disease, which is very prevalent in bordering areas of US. But the behind the scenes, it is the mathematics that is very relevant, how you capture those reality and how do you study it from mathematical tools. So, I mean, we all know there has been, it causes a lot of mortality, and we are worried about COVID-19, but the mortality is actually vector-borne disease far exceed. For example, just to give you malaria, deaths annual are much higher than in some parts of the countries, in some countries, than COVID-19. And so, but we never hear on a regular basis of these mortality rates. This belt is very, these are disproportionately affected countries like Africa, South America, even South Asia. India is part of South Asia, Southeast Asia. These are poor and disproportionately affected, not basically because of the geographical climatic factors, but primarily because of resource limitations, for examples. So that's why, well, I worked on many of these diseases. This is NTD stands for neglected tropical disease. The word neglected is because there is very few resources that are given, but less attention from pharmaceutical companies, and much more deaths and mortality and morbidity from these. And these categorized, WHO have categorized these things into two types of diseases. Microparasites, I mean, actually there are 10 diseases that they referred it as, actually not 10, 21 diseases that are considered as neglected tropical diseases, and they are listed here in terms of viruses, protozoa, bacteria, and helminths. Helminths are the worms, for example. So my focus has been on the disease that are circle red, like Chagas, leishmaniasis. I worked on dengue and chikungunya. Lymphatic filariasis, I was part of the field trials for lymphatic filariasis and soil-transmitted helminths. Even schistosomiyces also, I've been working on it. But primarily, I am focusing today actually on Chagas and leishmaniasis, which is protozoa-borne diseases. Well, some scales, when we talk about some scales that are, of course, is caused by there are different populations that are interacting for this disease to spread, such as pathogen, vector, and also host, and the transmission route is very important. How is the transmission spread? Is it by biting of the insect, or is it by the animals eating the insects and therefore the transmission, or mothers to baby? So there's different types of transmission routes. Not only that, there are different types of hosts also available for this type of diseases to spread. So it becomes very complex very quickly if you truly go into ecology of the infectious disease in that regard. So just to give you an idea, for example, there is an urban cycles you can talk about. You can talk about domestic cycles where Animals such as goats, buffaloes, those which are domestic animals can participate in the cycles. But then there could be zoonotic cycle, that is wild cycles, for example, sylvatic cycles, where wild animals and wild species are involved in it. So it depends on what you are modeling, and the model is very different, whether you are modeling an urban cycle, or you are modeling in a peri-domestic cycle, or you are modeling in the forest or wild cycle, for example. So just to give you this brief outline on it, again, as I said, there are three populations that drives these vector-borne dynamics. But not only that, the characteristics of these populations are very critical. It's like for vector is, for example, mosquitoes or sandflies. These are responsible for the disease. The feeding preference, how do they feed on different hosts? Whether they prefer one host versus other host, change the dynamics of it. And that is very critical to understand the biology of the vector-borne diseases. Otherwise, we are just doing one of the exercise from the book, if we are not really truly understanding the biology in that thing. Host species, there is animals, as I was saying, involved. There is different types of animals. Even avian birds are involved in as a host for West Nile virus, for examples. So host species are very important. Host behavior are all for whether the host move a lot. For example, if you look at in Bihar, the population is mobile quite, I mean, they move in a short span of time for work related or for different reasons. Whereas if you take in other states, it is not as much movement as compared to Bihar, for example. And then the transmission cycles, what are the hosts that are responsible for pathogen to circulate in the infection? So these are all makes it very complex to understand someone. And I will give you an examples of how the complexity result to a different implications for the results, for the mathematical results. So well, scales have started from physical aspects before that it is defined, but the physical space scales when we talk about is defined in a 2-dimension, which is space and time. Whereas when we talk about ecology, there is more than 2 spaces, and the third space could be the size of an animal. If the bigger the size, for example, larva pupa are not responsible for the infection to be transmitted, but adult vectors are responsible for the infection. So size of the animals or the stage of an animal is responsible for the infections. Also, it could be habitat conditions that could be responsible for the infections, or the higher density of the number of people in the community could be also responsible for the higher or lower infections. So some of the scales also is in the how the disease is transferred. Is it transferred from mothers to babies, or it is transferred from bites of the insects, or it is different species has a different influences. So scales are as very important. Now, why is this scales I'm talking about? Because when we come down to intervention, so one of the major reasons why governments are not talking about mathematical models for COVID-19 is because we are far from reality. We are far from reality in many cases, and we have been way off in that. And so the reason is, when you talk about the scales, the interventions are scale dependent. So which scale you should put the intervention becomes very critical in vector-borne diseases, for example. So it is not like all size fit, one size fit all, basically. This is our work that we did a long time ago, I think in 2017, where we are distributing things on a scale basis. So climatological framework, that is one scale when you are talking about climate and it is changing the density of the populations. And the models are very separate, the data is very separate, and implication coming out of it is very separate. Then ecophysical, where we are talking about life stages of different sizes of insects, so larva, pupa, those things. And the models are very different than human incidence. So these all scales are, of course, connected, but they are usually what we do is we model it one at a time. Epidemiological stage, the stages of the epidemiological stages is very important. Interventions are different at different scales, for example. And then when we come to treatment and diagnostic methods for any of the infection, then the scales also is within the body. And that is at the cellular level that we are talking about. So scales becomes very relevant when it comes to the modeling of this thing. Now, again, as I said in the starting, giving broader perspective of complexity and how do you handle multi-scales rather than going into one study. These all studies are online that I will be talking about. Another complexity comes into pictures. Different types of hosts are there. For example, accidental host, they get infected and they get symptoms, but they don't transfer the infections to the other, or they don't have a multiply. The infection never gets amplified inside the body of the host. So those are called as accidental hosts. There are incidental hosts. That means they are bad transmitter of infection. They will not develop the, see, in the host, the pathogens going to develop in the stages and they don't get developed, and those are called incidental. Primary hosts are the one where they can multiply, but they can affect the host itself. That means they can kill the host, for example, primary host. But they will multiply. They will multiply inside the host, and they are called as primary host. And reservoir hosts are the one which are the best one from the host, from the pathogen perspective. They can harbor the pathogen for a long period of time without impacting the host itself. So there are different types of, and what you model changes the equations or changes the functional form of the terms that we are talking about, interaction terms or the nonlinear terms that we use in these type of models. So just to give you an example, okay, let me see what happened to my, yes. So what did I do here? So there are very common terms that are used in ecology of infectious disease. Those are called dilution and amplification effects. Basically, what it means is whether if there are new hosts are coming in the transmission cycle. For example, if the insect starts biting a different animal, and so there are more hosts now. That is responsible for whether that will going to increase the infection in the community or decrease the infection. So by default, you will think that it will increase the infection because there is a new host, but that is not always the case. And that's why there is a dilution effect. So also, even if you increase the host, you could still have a dilution of the infection. Infection can come down in the prevalence. And that's because of the vector preference. There's something called as vector preference. And it is in this term. Can you see my, by the way, the cursor, right? I hope that you all can see my cursor. Dilution is basically the alpha V is a vector preference. The vector prefer, what is vector preferring? Infected host or susceptible host, for example. That could be the preference of the vector. Or vector could be one species of the host versus another species of the host. Host competence, on the other hand, is another terminology, which means how is the infection get transfers from the host, whether they are good transferable or it is bad, because the vector, the pathogens are developing in stages and inside the host. And whether the host is helping the pathogens to grow or whether it is decreasing, that becomes very important, and you model those things. So amplification means when you introduce new hosts in the populations, see, there's nothing like a statics that these are the host of the infection and it will remain always a host. Things changes. Nature is very complicated and it doesn't remain static as same all the time. So there's no host starts to come, and the infection may amplify because the new host that is joining in the infection cycle might be a good transmitter of the infection. If they are good transmitter, then more host is amplifying the total infection in the population. But if the new host that is there is not good transmitter of the infection, they are not encouraging the infection to grow within the host, then the total infection will come down and it will dilute the total infection. So even though more hosts are coming into picture, but that is not always increasing the infection. So increase in host species could also different types of host, if the density of them will increase, if the count of them will increase or abundance of them, then also the likelihood of epidemic might reduce or increase depending on the situation. Okay. So these are the basic ideas on this. This is a simple caricature of a vector-borne disease model where you have vector population. There is human population is in the bottom, and then there is host 1 and the host 2. By the way, this is for West Nile virus I've taken, but this is just a general caricature of it. And if you can see that vector preference is coming in this A1, A2. And then in here, it is a host competence. Different hosts are transmitter of infection in a different parameter. Different parameters are used. So this is how basically we capture something like that. It can be captured in a more complex way, but this is just a simple example. So the aim of my total studies have been basically looking at how host diversity, incorporating different types of hosts, changes the amplification or total prevalence in amplification and dilution effects. And how are the different factors, different mechanisms of ecology of infection, like vector feeding preference, host competence, how good is the host to transfer, transmission routes, how the infection is by the bite or by the mother to babies, how is the routes important here, whether the movement of the people can play an important role in that host in the amplification and dilution factors, or the count of the vector and a host species important. Even the epidemiological states, whether in some places people will only be asymptomatic and they don't develop a symptomatic infection, whereas other and people travel other place, people could have both asymptomatic and symptomatic, and there might be a movement between these things. So that will disturb the balance of the ecosystem, and therefore the complexity will be very critical in that case. So the first study I will be talking about the scales related to host lifespan. Different host has a lifespan, and the scales are related. For example, mosquitoes live for 15 days, whereas humans live for 60 years. So the scales are very different. Epidemiological scales are very important also. So let's talk about epidemiological. This is the first study that we are doing with Rajendra Memorial Institute of Medical Sciences in Bihar. This is on leishmaniasis. They have been targeting with the help of WHO for quite long time to eliminate this disease. And we have been working many studies, but today I will be talking about the non-adherence people, non-adherent to the treatment. Those group of people, actually the treatment of the leishmaniasis, this is on a leishmaniasis disease, all called as Kala-Azar, lasts for 28 days. And people don't, and this you have to go to the hospital and take the parental treatment, and that's why. So what happens in the surveillance, when it's the first day of the infection, they count the person coming for a treatment, but then they don't count for that the person is going to complete the treatment or not. Treatment is 28 days. So if they don't complete, then they go in the population and spread the infection. So you can see that importance of treatment non-adherence. If they don't complete the treatment, their parasites in their bodies are there, and therefore they can transfer the infection. So the question is, so what we have identified is we wanted to quantify, is data relevant to the dynamics? And so we used a simple models, human vector models, to study the role of treatment non-adherence. And so the data said it is around currently, so this is the left-hand side, you see the distribution of non-adherence from a clinical study. And there is and we found it is 10 days. It is 10 days, people on an average, people stay in the treatment, not 28 days, which is a full length of the treatment. They live for 28 days. And so what we found is the reason it is the disease is not getting eliminated. So 2010 not eliminated, 2015 not, and now 21 they are trying to eliminate, which will not be eliminated because non-adherence is around 10 days. And our model estimate says in order to eliminate the disease, you need 17 days or at least of treatment on an average for a population in order for this to eliminate. So this is important, and we studied some analysis on the role of non-adherence in that. Now the question is, how do you model non-adherent? I mean, one way to model to say the T is the treatment is an infected compartment, you just send some certain portion of the people to the infected compartment, which is what is non-adherent case, because if they adhere, they will recover fully. So if they don't adhere, then they will go to the I compartment and spread the infection. But their impact is also coming in this infectivity, whereas they are going in the population and infecting because they are not completed their 28 days of treatment. So this is one type of model, but that is not the only model, by the way. So let's study first of all, how do you analyze this model? So there's we have used a single perturbation theory to differentiate between the scales. Here the scales are, we have taken this epsilon, which is basically taking into the average lifespan of humans and average lifespan of the vector. Vector live for maybe, vector is an insect, sandfly here, and sandfly live for 14 days, roughly speaking, adult sandfly. And humans live for like 60 years, roughly. So epsilon is very small quantity because denominator, we can take it very small as compared to, and we can, the reason we want to do this singular perturbation and reducing the scale is because we want to simplify. This system is still complicated to do much of maths there. So, so we can use this standard theorems in the literature, Levinson and Hoppenstedt, to reduce the system by taking the vector population at the quasi-stationary state. And therefore you could study a simple model and analyze it, again, reducing the scales from vector host model to only host model, for example. Now, again, I was devoting my time to, there are many models for how do you capture treatment non-adherence. And we always struggle about it. So we modeled it in another two different type of model. And we wanted to see if these models are all same in some sense, what models we should pick. Usually what we do is we start with a model and analyze it, but that is not the best way. So you have to try. So left model, we used it because it's called a linear chain trick. And basically what it is saying is, instead of exponential waiting time, you have a Gaussian waiting time. And therefore, you are using two sets of data, mean and standard deviation, rather than one set of data. And other model is a very simple model where you have temporary recovery. The VH is the people who are temporary defaulted the treatment, and CH is the people who are completely taken the treatment. So VH will going to infect now in the infected stage, and that's another way of capturing it. But remember, there is a cycle here. The people will continue the cycle of infection. So how do you compare these models, results of this thing? So we took some kind of understanding to reduce this model in some ways. And I think I may have few minutes, right? So I'm not keeping track of the time. But this is the first model. So the way to reduce this model, the one on the top right, is by combining this all treatment. By the way, T1 stands for, let's say, first day of treatment. T2 stands for of the second day of treatment, and so on and so forth. So I'm just taking an example here with a 3 treatment compartment only. And we want to make this thing in a reduced model, in a simplified model. So if we combine these models, so we can compute, we can do some simple algebra to talk about average waiting time in the T hat compartment. This is a T hat. It's an average of this compartment itself. Although the parameters will change, these parameters will change here. This and this parameter will change, which will be a function of all these parameters that are. So we did some study which kind of relate this models to this one. But then also this parameter, which is related to the infectivity, will also change here. Because now there is one compartment instead of n compartment or 3 compartment in this examples. So, but doing that, in order to simplify this thing and make sense out of the model, you need to take into account the reproduction number should be preserved in this case. This is what our assumption is, and thereby preserving the endemic level of the disease. And so these two things, we are preserving between the model reduction, and then you can reduce it in a systematic way, in a right way. This model, on the other hand, the second model, could also be reduced where we are combining TH and VH into T compartment, and that's the most we can reduce. And therefore, in the same fashion, but remember, in this case, the cycle of this is an infinite cycle, so it's a series. And how do you capture the series and sum it? It's basically, this is the sum of that cycle, basically, average time that is spent in each compartment. Again, the same idea, the preserving endemic levels in order to scale it down to a least compartment possible. So again, so we came down to the lower level for both the models, and what we have found is some of the similarities and differences between the models. The meaning of the compartments are very different. For example, in this case, first one, IH consists of newly infected as well as treatment non-adherent. IH include this. T hats is a completely adherent people. Whereas in this case, IH is the only newly infected, and T is the treatment non-adherent. So there is different types of scaling led to a different meaning of it. So if we do this model versus that model, the results will be completely different, and the meaning of them is completely different. Now, again, each of the models has different meanings in that circumstances. I just wanted to give you model reductions ideas for scaling from a scaling perspective. Now, this is a scaling of the epidemiological stage, by the way. In the second study, I would like to talk about biting heterogeneity and spatial heterogeneity. So this paper just published into Open Science Journal, and this is talking about, now, dilution and amplification effects have been studied quite a lot in the literature. But the things that have not been studied is the role of amplification and role of different biting on an individuals, the biting heterogeneity and movement heterogeneity, how people move between spaces on the amplification and dilution. So, and we have derived some kind of threshold density on the sandfly population and reproduction numbers on this one. Worth employing it. It is very interesting paper. It is talking about threshold quantities of different types. And again, it's a vector host models with two vectors. One is reservoir A, reservoir B. But the important thing here is we have incorporated many heterogeneity, but the two new heterogeneity which has not been incorporated in the literatures is a part of this work, which is biting heterogeneity and spatial heterogeneity. How did we do biting heterogeneity? Basically, what we are saying is number of bites on an individual is actually, let me see if I can, yeah. So there are bites on a humans, and reservoirs are now random variables rather than deterministic mean value. And we use these ideas to, the infection rate is not like a standard incidence here, but it's a very complicated, which is basically a function of Poisson and gamma distributed random variables. And therefore, it is much more highly nonlinear in nature than usual standard incidence models in the literature. Well, again, this has no movement right now, but then if you incorporate movement, movement is incorporated as a short-term matrices. Here we are assuming the humans are moving. H means humans, A, B are reservoirs. The humans are moving, and therefore there is a summation here. The infection is now sum of all the people who are in a different populations. These are discrete populations, discrete patches. Space is the discrete patches, and A, B are not moving. And so the question is, this is incidence terms, lambda is an incidence term, is very complex. So if you can see that simple vector-borne models to this complicated incidence with a log term in it, and then the movement transition residence time matrix P is incorporated using the sum here. Okay, so just wanted to expose you that. Another study that we talk about is scales in pathogen populations. So this is a study in Chagas disease, which is spread by Trypanosoma cruzi. So we are modeling this border of the Texas, where there is one strain in...
Sir, can you compile your work within 5 minutes?
Sure. Thank you. Okay, so there are 3 regions here, but the cycles are very different in this. Cycles here is different. That means different host, different vectors are there, and cycle here is, and in the middle portion, the cycle coincide, the transmission cycle coincide. And the question is whether one strain will going to overpower what are the conditions under which one strain will overpower. And we did a bifurcation analysis on different strains. There are 2 strains in each region and 2 vector species, 2 host species, for example. So this is one thing. And actually I had a work on a stochastic process also, which is more theoretical. Here we are using functional law of large numbers and functional central limit theorems for a demographic stochasticity. But we are using a scaling of populations in that regard. Now remember, in a stochastic model, the population, the N, capital N, whether it is humans or vectors, is it a closed population or not a closed? If it is not a closed, then it's a random variable, by the way. And it's a random variable, it's much difficult to do the problems. And we are assuming that it is not a closed variable, therefore N is a random variable. And therefore, not a random, sorry, stochastic process, it's not a random variable, it's stochastic process. And therefore, it becomes very difficult, challenging to analyze the system. And we are talking about limiting form of this type of stochastic vector-borne system. Simple idea, but very important idea. So I want to thank everyone for inviting me. And I hope you guys have got some flavor of it. And I will be happy to circulate some papers that I have been doing actually. Thank you.
Thank you, sir. Actually, there are many questions in the